So it's always a challenge to give a talk when you're supposed to be moderating, and also to draw the unfortunate straw of being the very last talk in the session on the very last day. But Megan asked me to kind of round out this session by introducing at least some thoughts and directions we were planning on at UNL, in part because of a USDA Sustainable Ag Systems grant that we got about a year ago, actually focused on energetics in both beef and dairy cattle. So I'm going to start with something that regardless of who you are, you've seen this before, this traditional P equals G plus E, but I've complicated it a little bit. I've added this other small e on the end here. So the phenotype comes about through the sum of genetic merit, environmental effects that we know about, and I think that's the important delineation there. The big E is things like contemporary group, but then there's the little e too, which is residual, and we don't know what's driving these additional differences in phenotype. So it's something that's unfortunately unmodeled. And so then just what would seem like a very mundane review of what heritability is. All too often at industry presentations, somebody like me would say heritability is genetic variation divided by phenotypic variation. And that's probably what we see in undergraduate textbooks, but that's slightly misleading, right? It's additive genetic variance divided by, if we go back to this very simple model, it's really additive genetic variance plus residual variance as a denominator. We call that then phenotypic variance. Now, this seems like just an academic discussion, but the reason it's important is if I can take this very basic model and actually shrink the size of the small e, okay, the residual, then by doing so, I can increase the estimate of heritability, right? So if I can condition out more residual variation from whatever I'm trying to model, I can get larger heritability estimates, and by default, then more accurate estimates of breeding time. So the questions that I want to pose today, and I'll take you through some answers along the way, is with respect to methane, how heritable is that? We've heard some at this conference already. I'll provide some estimates that are beef cattle centric. Then what degree does the microbiome contribute to enteric methane production? What degree does the host, so additive genetics, control the relative abundance of features of the microbiome? So if we review those first three questions, how heritable is methane? How much does the microbiome control enteric methane? And then how heritable is the microbiome itself? Leading to the last question, does adding metagenomic data aid in both phenotypic and genetic prediction of enteric methane? So these are estimates that I crudely pulled from a nice summary by Dressler and others that was published in Translational Animal Science in 2024. Methane production, that's how much methane an animal produces in a day. Methane yield, that's methane produced per unit of feed intake. Methane intensity, that's methane per unit of animal product. And then residual methane is akin to the way you might think about residual feed intake. It's really a restricted index. And these are average heritability estimates that I pulled from that review paper. And the take home is moderately heritable, right? So regardless of how you define this trait complex, it's moderately heritable. So akin to what we may see for something like average daily gain or weaning weight, things of that nature. So no doubt we can make genetic change to any of these if we place selection pressure on them, although I would argue some of these probably lend themselves better to genetic evaluation than others do. I'm not particularly keen on using ratios as something in a genetic evaluation. They tend to be unstable. So there very well could be a difference between the thing that we evaluate for genetic prediction and the thing that industry might use to benchmark or to advertise product. Okay, there could be differences there. Now, what about the contribution of the microbiome to methane? I pulled out just two papers, and somebody in here from academia can say, well, I published one, and it's not an exhaustive literature review, okay? But here's a paper from 2022 where the authors reported 29 rumen microbial genera that were heritable and genetically correlated to methane output. And then in 2018, different than others, attributed to differences in methane production, with 13% of that coming from host genetics and 13% of the differences in methane being attributed to the microbiome and 21% due to host genetics. So it is both of these sources of information together contributing to the differences that we see in methane output of animals. Now, the trick, the tricky part, I guess, which I'll get into a bit later, is these two things are not orthogonal, right? So, given that the host also selects through additive genetics for what the microbial composition is, these aren't two independent sources of information. And then lastly, I understand this is hard to see, so I can tell you whatever I want about it, and you're forced to believe me. But there have been a multitude of studies across a multitude of species that clearly show that the microbiome, however we wish to define that, is at least partially controlled by additive genetics as a host. And this really goes back to work decades ago in model species like mice, where that's been clearly illustrated, and now we've seen papers, of course, in beef cattle, sheep, goats, swine, et cetera, et cetera. And so what I have on, I guess would be the kind of lower left, we see heritability estimates, whether it be the OTU at species level, phylum or family level, and the mean or median heritability estimates are about 15%, right? So this is from the rumen microbiome. If we go to those line graphs on the right, that's actually ocular microbiome, and that's diversity, alpha diversity of the ocular microbiome at 4 different time points in the animal's life. But the first time point was kind of a naive, undisturbed ocular surface, and there the alpha diversity metrics were between 15 and 20% heritable. So plenty of evidence in beef cattle and elsewhere that the microbiome and certain individual features within the microbiome are heritable themselves. So just another example of that, moving to metagenomic data here, I have some details about what we did. This was Illumina short read sequencing, and there was quality control as what QC is to remove both human and bovine reads. And I'll get back to that QC for removing bovine reads, because when I figured out you're throwing away bovine sequence reads, I became really interested. The microbiome features that we used are open reading frames, ORF. Don't worry if that doesn't mean anything to you. I'm still struggling with what that means to me. But what it really is getting closer to the function of the features in the microbiome beyond just OTU or species level. And we wound up with about 16,580 of these open reading frames, again, features of the rumen microbiome left for downstream analysis. And what we did is we got the relative abundance of those and then log transform. So this kind of gives you a sense of how heritable that was, and this is across about 700 beef cattle. These were actually U.S. marked animals. You can see about 10,000 of those 16,000 ORF had heritability estimates that were non-zero. Obviously, a density pretty close to zero here, but still several of the features, at least in this particular study, functional features of the rumen microbiome that were under some degree of additive genetic control, and about 100 of them were pretty heritable. So heritability estimates of about 40% or more. So if I just kind of check in for a minute summary here, methane emission by ruminants is heritable, and we've heard about that for the past couple of days. So we know that we can argue whether the estimate's 15% or it's 25%. That's all nuance, okay? It's heritable. The microbiome contributes to methane emissions, and that makes sense, right? I'm not a rumen microbiologist at all, but I know there are these things called methanogens. And because of that, it's reasonable to say that the microbiome contributes to differences in enteric methane production. And lastly, the microbiome itself, and here we're specifically talking about the rumen microbiome, is at least under some degree of additive genetic control. So that brings us back to this very basic model. So now that we're thinking about modeling methane, that's the phenotype of interest, we still have this big E part, environment contemporary group, which of course accounts for differences in the diets of the animals, and what other systematic effects that we think appropriately model. We have genetic merit, but it's genetic merits of the animal for methane, but it's also the genetic merit of the animal for the microbiome that's in its rumen as well. And then we have this little e out here, the residual, which I would contend part of it could also be the unmodeled microbiome of the animal. So with that, you can think about expanding this basic model with more letters. So the phenotype, in this case for modeling methane, becomes a function of genetics of the host, that's G, environment, the environmental things we know about, that's the big E. M now I'm using for microbiome, and then G binding, so the interaction of host genetics and the rumen microbiome. And then we still have the little e residual on the end, but hopefully the little e proportionally not as impactful as it was before, now that we actually implicitly model or explicitly model the rumen microbiome. And so an example of this for a related trait complex, Dr. Drew Lucampus, he was a PhD student of mine, now a postdoc. I heard him introduced as doctor yesterday, and it just kind of struck me as funny, but it's technically true, I guess. And I hope he doesn't expect me to call him doctor when we get back home to the office. That's not going to happen. But this was his PhD work, and these are USMR GPE animals from two different diets. Steers were a high concentrate diet, heifers high forage diet. Those animals that were then split up within diet type into some different implant regimes. So that's the NI, no implant, HI, a high implant, MI, moderate implant. And then at the bottom of this, you'll see different year-season combinations, right? So these animals were split up a considerable number of ways, but that's the way the study was designed, if you will. So what Dr. LeCamp did was ask the question of how might we go about actually modeling jointly host genetics and the metagenomic effect. And so we fit a genomic relationship matrix that would be standard to those that have done those kinds of things using the first method of rating. The microbiome did it, a microbiome relationship matrix is called, which I really dislike that phrase. It's a covariance matrix based on functions of the microbiome. But form that either doing a similar method as what we did for the host genetic component, or did something similar to what Elizabeth Ross did in 2013, which is really analogous to the second method of Van rating for those that are familiar with that. And then took those metagenomic relationship matrices and either weighted them based on a function of the heritability of that microbial feature, or as I'll go through what I call a multi-population kind of structure, given that animals are either on a high concentrate or high forage-based diet. And then the models fitting either a genomic component only, a microbiome component only, additive, which was both genomics and the microbiome component, or then an interaction model where we fit the animal genetic component, the microbiome component, and then the Hadamard product of those two to get the interaction. So some of you in the room would recognize the name Dale Van Vleck if I said it, legendary person in quantitative genetics from the University of Nebraska, Lincoln. He just turned 92 yesterday. And I'm reminded of that because Dale always told me that there was no need to show equations in presentation. Because he said the people that are going to understand it will understand it just fine by me telling them. And the people that aren't going to understand it any better if we put the equation up there. But I did listen to Dr. Van Vleck, and I put an equation up here. So what I have here is an example for the weighted microbial relationship matrix. So this M sub VW is following the first method of Van rating. S is just a matrix of rows or animals. The columns are the ORF, those microbial features. P, we're just centering it. So P has the mean relative abundances. And then in the denominator, we're simply scaling this using the variances of all those ORF. The D matrix, that diagonal matrix, that's what we're using to weight these microbial relationships, if you will. And that's 1 minus the heritability of the microbial feature, right? Because what we're wanting to do is create as best we can independence between the additive genetic effect of the animal and this microbial random effect that we're fitting. Okay. On the bottom, I have MRW. This follows kind of the second method of Van Raden, where these seed matrices are centered and scaled, but they're scaled by each column independently. Okay, instead of the first method above, where we're dividing through by the sum of all those variances. So these are really the two different weighted microbial relationship matrices. Then I mentioned we had two different diets, high concentrate, high forage. You look at principal component or PCA plot of the rumen metagenome, and it clearly clusters into two groups. The groups that were fed high forage, and the groups that were fed high concentrate. To any nutritionists in the room, that probably makes great sense. But that got me to thinking, there's great theory in the literature to deal with multi-population genomic relationship matrices. And so why not utilize that same kind of methodology here, when clearly we have two distinct populations in the rumen metagenomes. And so that's what this is. This is the metagenomic relationship matrix following what Mario Callas's group did in 2017 with the multi-population genomic relationship matrix. So now we've got these two different subgroups, largely the ones fed concentrate, the ones fed forage, and we're scaling those differently. It's basically what we're doing. Okay? So that's what we call the data-driven approach. Looked at the data to see how they clustered. We considered those different populations, and we took that into account when we formed the metagenomic relationship matrix. We went through and did validation of these different things too, randomly, and then what I call LODO, because I think it sounds cool, but it's basically leave one diet out. Okay, that was the cross-validation strategy. What I want to take you through is results here. So you'll see heritability, and the take home is when we begin to fit the metagenomic component in the models, we get higher estimates of heritability. Now you look at these and say, well, in some cases that increase is negligible, but it went up like we expected it to because we conditioned out residual variation. That's why the heritability went up. The M squared is when in the literature they call the microbiability, which that's another phrase I really dislike. But somebody used it once, and so we're stuck with it now. One of these days I'm going to say that in the room where the original author's in, I'm going to get in trouble with it. But it's really the proportion of variation explained by this metagenomic random effect. The S squared is the sum of host genetic and microbial effect. And then the I squared includes the interaction. And so again, three main takeaways from this: heritability estimate goes up as we fed the metagenomic component. We're able to explain more total variation when we fit both the host genetic and the metagenomic component. So again, that's the S squared value. The interaction depended on the way we form the metagenomic covariance matrices as to whether we actually increase much in that interaction. So the take home from a related trait complex, and what I just showed you was results for average daily dry matter intake, so very much related, as we heard before, to enteric methane output. And the take home from that is additive and interactive models explain more variation and had increased prediction accuracies as we predicted the adjusted phenotype. And so contemplating host genomics and metagenomics jointly seems like a reasonable approach for the trait complex of methane. A couple of caveats with that, though. The microbiome, if you look in the scientific literature and you listen to scientific presentations, the microbiome by some might be considered as an additional random effect, as we've done here, or as a correlated phenotype in a kind of multi-trait approach. So in other words, a phenotype or features of the microbiome, and you're actually getting breeding values or EPV for those features of the microbiome. And as I said, the literature is kind of mixed on that approach. Regardless of approach, I would say that right now, shotgun metagenomic sequencing isn't necessarily cheap. And so as a consequence, we've got to be cautious of chasing down an indicator trait that's actually also very expensive. It's also a bit unclear to me if we consider this as a correlated phenotype. What is this phenotype exactly? because we've got an opportunity for tens of thousands of features from the rumen metagenome. So that becomes a bit complex, and you've also got to consider that there are relative abundances. So if I change one, by design, by definition, I will change a whole bunch of others, right? So I think we've got to be cautious in what we're actually selecting for if we go down that path. So to the current effort, somebody more creative than I did came up with this slogan, "The cattle methane in you." There's a play on words there. It took me a long time to understand it. For those of you that are football fans, there's the old joke, what's the N stand for on the football helmets? Knowledge, right? So for any smart ass in the room that we're gonna say that, I beat you to the point, okay? So the N here is for knowledge. For anyone that doesn't get that joke, knowledge has a silent K in the front. But you don't have to admit it if you didn't understand that. But this, again, is a USDA SaaS grant that actually Paul Karnoff, dairy nutritionist in our shop, was awarded about 12 months ago, and really focused on energetics. So methane is just a component. And this work really was spurred by an initial project that I helped Paul's grad student Addison Carroll with, where Paul had 530 Jersey animals. So he'd gone through his respiration chambers and done these energetic studies on. And he asked me, "Do you think there's among animal variation for these things that we're mentioning?" And I said, "Yeah, there is." And then I asked, "Well, what are you measuring?" And so he was measuring things like feed efficiency, methane, and then these ratios like digestible energy to gross energy, or metabolizable energy to digestible energy, net energy for lactation to metabolizable energy. And you'll see the kind of what I call yellow to oranges is the among animal variation. And it's sizable for all these things, in particular for methane. And so that convinced my dairy nutritionist friend that, hey, there's something here with respect to genetic control of methane, but then also for some of these more refined energetic phenotypes that he was interested in. So this is the project team here at UNL. I'm not going to list all the names, but it's diverse in terms of expertise, dairy nutritionists, feedlot nutritionists, people in the precision livestock world as well, with Tammy Brown Randall, Simona Fernando, who's our rumen ecologist at UNL, very talented one, and then at US Mark, Jim Wells and Brian Neville as well. So our aims in this are identify host, microbial, environmental, and management factors that influence methane production in ruminants, and then to develop some science-based, obviously, strategies for dietary influence on the microbiome and genetic interventions to reduce methane emissions. And I'll go through a few things that we're specifically going to do. All these animals are going to go through what I call head boxes. Those are respiration chambers at UNL. There'll be 560 animals total across both beef and dairy. The majority are beef cattle animals, and they're animals from US Mark's GPE population. And so this project is really seeding, in a lot of ways, a project that Bailey Eagle's probably talking about right now, that's going to ramp up the capacity to collect these kinds of traits at US Mark. So through this, of course, we'll genotype the animals, do shotgun metagenomic sequencing of them, and go through the traditional, what I call MLOS, so this kind of microbiome-wide association studies to identify more informative features of the rumen microbiome. But what's really exciting to me is all the, what I call phenotypes we're going to get. So not just gases, but also things like digestible energy, metabolizable energy, net energy for gain, because we're also doing fecal collections and urine samples as well. So a limited number of animals, but I think a lot of really novel phenotypes that we're going to collect. And one other thing, Paul Karnoff, when he talks about this, mentions this part as our moonshot. I don't like referring to it as the moonshot, but I think that's his way of hedging in case it doesn't work. But the moonshot, if you will, and we have a pilot project that's wrapping up on this right now, is actually taking a rumen sample from high methane emitting animals and a rumen sample from low methane emitting animals, and using those as inoculates then for newborn calves. And so we could take newborn calves, and you can see over here, in the pilot study, we used 6 newborn calves. And so within 24 hours of birth, we're inoculating a rumen with the microbial composition of either the high or low methane animal. And so what we do then is follow those animals through and ask basically 2 questions. That basically microbiome that we implanted in the animal, is it static? So can we recover what we inoculated them with much later in life? And then 2, do we see a difference in phenotype between those 2 groups? Right? So the idea is, and I know somebody's going to ask, how are you actually going to do this in practice on ranch, right? That's not the question right now. The question is, does this work? But the idea is that we can provide desirable microbiome very early in that animal's life, such that that desirable microbiome not only persists through the animal's life, but also manifests in a change in phenotype that we want. With these kinds of projects, data size is a problem, all right? And I think there is opportunity to improve genetic predictions and also phenotypic predictions to inform management decisions by contemplating host genetics and metagenomics at the same time. But to do that, we need both at scale, and so costing becomes an issue. All-in costing, one of the factors is we oftentimes rely on esophageal tubing to get the rumen sample. And I've never done that, but the people that have complained to me about how laborious that is and how we're limited in the number of animals we can do. Some of you have heard of doing oral swabs. I think there's sufficient evidence to say that the microbiome recovered from those oral samples is not the same microbiome as we would recover from a rumen sample, but that does not mean that the microbiome from the oral sample is not predictive of the thing that we're really interested in. So I'd ask that we not conflate those 2 things, right? The oral sample can still be very useful at predicting the trait that we're interested in. Timing of sampling is going to be important too. Getting a sample closest to when the phenotype we're interested in is expressed would be ideal. But if we want to use that single metagenomic sequencing event to cover a wide array of phenotypes, there might have to be some give and take there. And then my last comment is it would be nice to use a single biological sample to generate both host and metagenomic sequence. And just as an example of that, again, Dr. LeCamp, and this is actually not rumen, it's ocular, from a relatively small number of animals. But what I'll just take you through is that he looked at concordance after having imputed host sequence from the metagenomic sequencing event from those ocular swabs. We're looking at the ocular microbiome. And we had those animals already arrayed genotypes, so we used the arrayed genotypes as the truth. There's some flaw in that, but that's what we did. You can see that we had unfiltered data sets and some were refiltered where we required 75% confidence in the imputed calls. And I point your attention to the very bottom row here. That's where we filtered based on confidence in the imputed call. Animals had at least 0.1x of sequencing depth, and we were able to verify their ID. In other words, we could match them back to array genotypes sufficiently, because we did have some animals which were clearly mislabeled. Their metagenomic sample was clearly mislabeled. But when they meet all of those thresholds, you can see the mean concordance here of greater than 98%. So there's an opportunity to get a 2-for-1. If we're doing metagenomic sequencing, we could be genotyping those animals with the same sample. What we're looking at now is, well, how much can we reduce the metagenomic sequencing depth? So we're downsampling to see where that kind of ideal spot is, right? So how shallow can we go there to get both uses out of the same sequencing event? So the overall summary, methane trait complex is heritable and is correlated with growth and feed intake. I think that's fairly well established. The estimates will change over time, but we know those things to be true. Indeed, other countries, as we heard from Jason Archer, other countries have already moved forward with selection programs for this trade complex. In the US, I think we're just starting to really ramp up data collection and starting to think towards that. Selection in the future, and I didn't cover this, but just to throw it out there, we need to contemplate the economic benefit to an enterprise. So in other words, some kind of whole system selection index that contemplates methane. And I see a lot of grant proposals that say that we're going to put methane in an index. That's more complicated because there's not direct economic signal right now for it. So it kind of lends itself almost to this desired gains approach. How much do we desire to change methane absent economic signal? So I think doing that requires careful thought, but certainly just throwing a methane EPD out there and saying, now best of luck readers with what we do with it isn't the right approach of it. Okay, it needs to be part of the comprehensive decision-making process. Considering all sorts of variations of the genomic, metagenomic will lead to more accurate genetic and phenotypic predictions. And this new study I mentioned, the USDA SAS grant, will consider more than just methane. Really, we're focused on energy sinks using both a genomic and a metagenomic approach. So with that, I'd like to thank a great many people that helped with everything I presented. And then, of course, thank the funding model as well. And I'll be happy to take any questions. I'll be happy to adjourn as well. Yes, Cliff.
Have you guys done anything like structural equation modeling, trying to get directional causality of microbes to methane to host? It's really interesting, and I don't know exactly how it fits in prediction, but just curious.
So the short answer is no. I'm being a bit cautious here, because I'm aware of who all's in the room. But causality, when you think of the dimensionality of these data, and the fact also that by nature of the way, at least we and most other people transform, we create relationships among them because they're relative abundance, right? And I think that adds additional complexity in those two things together add complexity and inferring causality. There are certainly people that have basically considered it as an intermediate omics data, and they may call it something different, but it's really a set of hierarchical mixed linear models that are employed to account for both of these sources of information. And I think, at least looking at that, there's biological appeal to that approach. There's also potentially an issue of compounding error as you utilize predictions to feed into something else too. That's a long-winded answer to what was a simple one.
Thanks.
Jessica? So I want to make sure I heard you, Jesse. So that inoculant I mentioned towards the end, how much and how do we deliver it? Yeah, so these are like boots on the ground things that I have the luxury of being a guy that says, I like to do this. Can we? And then hope that the people that do it don't say no. No, so in terms of amount, it was less than 10 mils, okay, in terms of amount of inoculant. And what we did was use an actual drench gun to stick in the animal's mouth and drench them. We tried to do it as early after birth as we could, but we got them all done, I believe, I hope I'm not lying, within 12 hours after birth. That newborn microbiome is not formed yet, but it begins to form based on, of course, vaginal microbiome passes through the vagina, and then the environment it's around. So we thought the earlier we could get it in there and seed it, if you will, the better off we were. But that's approximately how much depth, and that's how it was delivered. Yeah. Well, we're that was sample from live animals, so, esophageal tubing, right, of live animals that were extremely for a trait complex we were interested in. Yep. So not altogether different than saying I have a fecal sample, right, in humans, and I want to do an implant. Yeah. Yeah, Shane.
I don't think I can talk about this into the esophageal.
I'm pretty sure you could.
So with the saliva swab, walk us through that process and collecting that data, where to send it.
Yeah,
Need a sample.
so do you need to sample all of them? Those are always hard questions, because I like to sample everything you can get, understanding that you may not process them immediately, but you've got the sample, right? So those can be stored. And normal swab, to me, doesn't seem that cumbersome if it's being tied with another working event, right? If there's, some kind of re-implant going on, something of that nature, maybe when they're placed on feed, those kinds of things. It would at least seem to me to be logistically possible to get it on all animals. But I agree, the esophageal tubing is, that takes too long in scale. And then you're limited on size of animal too. We've tried to do it on fairly young animals, Shane, and there's real risk there, of injuring the animals if they're not big enough. Larry, you have a comment?
Yeah, but when we've done oral swabbing, it's usually been, 15, 20 seconds per animal if we can control it reasonably well.
Compared to 4 or 5 minutes, if you're tubing them,
3 minutes with a really good person, yeah.
which I've tried to tell people who do that on smaller scale research projects, that 3 minutes times thousands in a day adds up really fast.
That analysis map, walking through where he's in that, and we'll do it.
Well, so we're doing that in-house right now. I know US Mark has the capacity to do that as well. So we're doing, for the grant I talked about and for the projects I talked about, we're doing that sequencing in-house.